Dr. Lee Gan. She's a professor and director here at Cornell, and Lee will talk to us about a brain-penetrant small molecule targeting innate immunity for neurodegenerative diseases. Lee?
Thanks, Jeff. What I'm going to talk about today is Aeton Therapeutics. That it has two scientific co-founders, myself and Dr. Subhash Sinha, and with the tremendous help from Jeff, Dr. James at the Weill Cornell Business Development Office. So microglia play a pivotal role in modulating neuroinflammation, which is not as a consequence, but a driver in the CNS injury, aging, and neurodegeneration. And the key cell type that mediating the neuroinflammation in the brain is microglia, which are brain's innate immune cells. And the signaling from the microglia and other cell types create this feed-forward mechanisms that can lead to, when aberrantly activated, it can result in neuronal and synaptic loss. And in Alzheimer's disease, it becomes very prominent role because of the enrichment of AD risk alleles that many of them modulating microglia function. And one of the key pathways that Aeton focus on is called cGAS-STING pathway. cGAS is an enzyme that senses double-stranded DNA in the cytosol, and then lead to activation of STING, and then activation of type 1 interferon response, as well as kappa B response. And many evidence suggests that this response is neurotoxic. And in particular, as illustrated in this slide, summarizing some of the findings from my lab, as well as from other labs, showing the cGAS activation is at the center of neurodegeneration pathway, and that promoting neuronal injury. So here I in the highlighted in red are the disease indications that my lab have been focusing on, that including Alzheimer's disease that were published in 2 papers last year, as well as progressive supranuclear palsy, frontotemporal dementia, ALS, Dravet syndrome, and TBI. Some of them are still unpublished. So from about 6 years ago, Aeton Therapeutics, with collaboration with my lab and Dr. Sinha's lab, have launched a drug discovery program to identify human cGAS inhibitors that are pre-permeable. So through this discovery workflow, we have identified 4 lead compounds, and we also have established an assay for target engagement using the downstream signaling, including phospho-TPK1. And in this published work, using the 2 compounds discovered at TDI, we did a proof of principle study showing that in the AD-related tauopathy models, this inhibitor is brain permeable and can improve memory function of these AD-related mouse models, as well as ameliorate synaptic loss. So this was published in 2023. We have also discovered that cGAS inhibitor can reduce the spread of tau pathology, which is well established to drive the cognitive decline in the Alzheimer patient. And you can see here that there is a significant reduction of the tau pathology spread when they are administered with a mouse cGAS inhibitor. This was published last year in Immunity. We have also moved on other neurological disorder models, as I indicated in the introduction. The ALS model, which is driven by mislocalization and pathology of TDP-43. In this case, this is a Q331K mutant ALS model. We find that the mouse cGAS inhibitor in this proof of principle shows it can significantly improve motor function, normalize metabolism, using the metabolic cage assay, a completely unbiased assay, as well as ameliorate the motor neuron degeneration that is caused by TDP-43 pathology. And this is all orally administered. We have also found, using human iPSC-derived microglia model that our proprietary human cGAS inhibitor can improve the phagocytosis function of microglia, human microglia, and dampen the interferon immune response. And in this most recent proof of principle in vivo study, we find our proprietary human cGAS inhibitor, 61-5, which is one of our lead compounds, can significantly dampen the tau pathology that is induced by seeding, as shown here. So moving on to another unexpected neurological disorder called Dravet syndrome. So Dravet syndrome is a childhood drug-resistant epileptic model. And in this study that is submitted for publication, we found that hyperexcitable neuron in the epileptic model can trigger cGAS activation, and cGAS inhibitor can ameliorate both the seizure as well as reduce the mortality of this epileptic model. So this is what we think that Aeton Therapeutics is hoping to do, is to start from rare diseases, and ultimate goal is to move this cGAS inhibitor to more common neurodegeneration models such as Alzheimer's. So there are several rare indications we're considering that including Dravet syndrome, PSP, ALS. These are all orphan diseases that can have a low barrier of entry and allow proof of principle in human. So this is a snapshot of the milestones. We have identified 4 lead compounds with nanomolar potency. They are BBB permeable with superior brain PK properties compared with our competitors. We have demonstrated proof of principle in multiple preclinical models. We have also established a humanized cGAS model, both knock-in as well as overexpression, for further testing of human cGAS inhibitors. We have an option agreement with Weill Cornell, and all of the studies I presented today were funded by non-dilutive funding of over $8 million from government foundations as well as other institutional resources. What we're looking for is the next stage to accelerate moving the cGAS inhibitors into human. We would like to be able to nominate clinical candidate within a year and complete IND enabling study with the funding of about $8 million. So we have PCT covers 1 series by Weill Cornell, as well as a provisional cover a 2nd series. And we have published widely on this topic, and we believe we are a domain expert in this field. And this is a team. I'm the biologist. Dr. Sinha is a chemist, assisted by Dr. Anjana Sinha, and also the financial matters are handled by Jeff Zhang, who is our interim CEO. Thank you, and I welcome questions about our program.
I have a question, but I couldn't figure out how to raise my hand, so I'm just going to talk.
Just go ahead.
Could you talk a little bit about your mechanism as compared to the cGAS inhibitors, I believe, that are currently in the clinic for inflammatory diseases such as lupus? I assume those are not brain penetrant, but would be curious how you compare your molecules to their biology.
Yeah, as you alluded to, that the Ventus compound is not brain permeable, and I think it will create a very interesting, we are eagerly waiting for their phase 1 clinical trial data to see the safety profile. So we are targeting CNS disease. So the differentiation factor is we have very, very good brain PK, which allow us to target diseases such as ALS, such as PSP, such as Dravet. So that's a differentiating factor.
Got it. So since it will be orally administered, the portion or the molecule that ends up going systemically, you might assume will be doing the same thing that the Ventus molecule is doing as well.
Yes.
Yeah.
So I think that's why I think their safety data would be very informative for us. But in terms of indication, I think CNS it presents a much more challenge as well as a great need for our aging population.
And I do have one quick question. So if I understand it correctly, you guys have done a number of proof of concept studies. And can you just kind of walk me through your path to IND? When are you guys looking at the IND? And have you guys decided which indication you are going to first file your IND in? Is it going to be the Dravet or is it going to be some other indication? Thank you.
Yeah, so that's extremely important question, and I would like to address this once we have the clinical nominate the clinical compound and in conjunction with medical experts for final decide which indication to go into. So to me, as a neuroscientist, as someone who has working in the neurodegeneration, of course, ALS and PSP would be something that I feel I have the network of experts that I can rely on to make the best judgment. But I think this is a combination of both the profile of the compound as well as the market competition. It's as well as a business decision. So I don't think right now we can make the decision for which indication to go into.
All right. Well, thank you so much.